I'm Maribel Rios at BPI TV. I'm here with Ashley Hesslein from Bayer Healthcare. Hi, Ashley.
Hi.
How do your project pressures on your biologics line affect your use of new technology?
Well, with new technologies, it's always a give and take. We have to be careful. So there's always a pressure and a movement trying to get us from some idea and research into the IND as fast as possible. And then if a project's going well, we need to move into phase 1, phase 2, and then launch as fast as possible. So we don't really have time to do a lot of customization for our antibody projects, and there's a big heavy reliance on platform processes. So what I'm saying is there's not a lot of time left over for a lot of creativity, a lot of innovation, and a lot of changes. There's always a pressure against those things. So to balance that, because we want to be making processes that are new and creative, we have to always have platform processes operating in parallel so that we can assure that our products are making it into clinical stages. And we always have to have backup options where we're trying something creative along the way. The second piece is that we have to have major changes. So incremental changes, small changes aren't really worth it typically. We usually only take the time for changes that make a big difference. So something like a resin with a binding capacity of maybe 5%, 10% higher may not be enough for us to be willing to jeopardize or put at risk any program. So we're looking for creative, innovative changes that make a big difference or solve existing problems.
Sure. As you increase the use of single-use materials, what new needs have come up?
So single use, as you know, has created a lot of benefits for the bioprocess industry, and there's a lot of major advantages that it has created, or a lot of advantages it brings to the manufacturing. But what has come up a lot more frequently is the use of animal-derived components.
Right.
So specifically, we are required to explain to the federal agencies that our materials are either animal-derived component-free, or that we have documentation certifying that the risk of TSE contamination is effectively none. So our raw material vendors have often supplied information with the single use because there are so many different types of plastics with connectors and tubings. They often provide documentation that the materials and the components comply with the EMA guideline 410, and they make a statement to that effect. Up until this point, that's been okay, but we anticipate that the vendors are going to have to start providing a lot more information, especially when we have products, as all our products will be ultimately in clinical trials or licensed outside of the EU or the US. What we need from vendors is better documentation for them to go back to their raw material suppliers, particularly with stearic acids and other bovine-derived components, where they explain directly how those were made and what conditions were used so that we can assure and have details around verifying that the harshness of those conditions reduced or eliminated the risk of TSE.
Very interesting. Thank you very much for talking to me today.
Yeah.
I'm Maribel from BPI TV. Stay tuned.